welcome to Edison Open House Global Healthcare 2021. In this session, we're going to be highlighting the work of cell therapy company Mesoblast, and I'm delighted to be joined by their CEO, Silviu Itescu. Silviu, tell me a bit about Mesoblast.
Sure, thanks for having me. Mesoblast is developing a suite of products based on several platform technologies that are derived from stromal cells, mesenchymal stromal cells. We harvest these cells from healthy donors. We're able to isolate them to high level of purity, and we take advantage of the fact that there's a common mechanism of action whereby these cells, through specific receptors, are able to bind to and be activated by inflammatory cytokines. And when they're activated by inflammatory cytokines, they release a number of well-characterized factors that are then able to modulate the immune system and regulate multiple arms of the immune system so that you end up with a durable anti-inflammatory response. That is a common mechanism, and our approach is therefore to develop a variety of products that target severe systemic or localized inflammatory processes where traditional approaches using small molecules or monoclonal antibodies just don't do well enough, and that provides the background to the various disease states and product candidates that we're developing.
So your main indication is chronic heart failure, so classic inflammatory condition. Tell me a bit more about that, because there's been a huge increase in the incidence of chronic heart failure, hasn't there?
It's a terrible epidemic in the Western world. More than 25 million people suffer from heart failure, more than 6.5 million in the US alone, and it's increasing. Look, it's a disease that's progressive. People progress inexorably from class 1, 2, 3, class 4, that's end-stage heart failure, despite maximal standard of care. And it is characterized by progressive and increasing intracardiac inflammation of the macrophages in particular in the heart, which secrete a variety of inflammatory cytokines, including TNF, IL-1, IL-6. In the last few years, it's become particularly evident that inflammation is at the core of this disease progression. And we've developed a therapy that targets the intracardiac inflammation, and through extensive preclinical work and then phase 2 trials, we were able to identify the appropriate dose of cells and the mechanism of action that allowed us to proceed to a phase 3 trial in 565 patients, randomized 1-to-1, targeting advanced class 2-3 heart failure, where we endeavored to inject a single dose of our cells by catheter and endomyocardially to see whether we could arrest or alter the natural history of the disease on top of maximal standard of care.
And you produced those results recently, and this is your drug, and I hope I've got the pronunciation right, rexlemestrocel. And tell us a bit more about those results, because they were, pretty amazing.
Yeah, we were very excited with the results. So the way to think about advanced heart failure today is that there are two main problems. One is so-called pump failure, which is dilated left ventricles are not able to handle volumes very well, salt overload. And the variety of drugs that are out there, ACE inhibitors, beta blockers, and a new generation, new classes of drugs, Entresto and the SGLT2 inhibitors, tend to address salt handling and volume overload, and have reduced generally hospitalizations due to volume overload. The major unmet medical need in these patients, however, is not that so much. It's actually ongoing severe progressive cardiac death and the complications of vascular inflammation, which is atherosclerotic involvement of the large vessels, heart attacks and strokes. And we were surprisingly able to significantly reduce both of those vascular ischemic complications, including cardiac death, in the patients who were at highest risk for this. and in a way that has not been able to be achieved with other small molecules.
Tell me about the difference, though, between the data for MACE, which was, I think, was your previous trial, compared to the data that you've recently announced.
Sure. So in the phase 2 trial of 60 patients, randomized controlled study, the objective was to identify the most appropriate dose going forward and to identify the appropriate target population as well as the therapeutic benefit. And so in that 60-patient study, which was published in Circulation Research in 2015, we saw that the highest dose, the 150 million cell dose, allogeneic dose, gave us the greatest benefit both in terms of the surrogates, volume changes and 6-minute walk, but most importantly, over a 3-year period, prevented any events, and the events were defined as a composite of either recurrent hospitalizations or cardiac death. And that's a traditional endpoint that many companies use for their agents. The data were not, the number of patients were not large enough to be able to decipher between hospitalizations versus cardiac death as an endpoint. So the composite was what we looked for, and that was the endpoint used in the current trial, the phase 3 trial. And it's a traditional endpoint to look at reduction in hospitalizations. We were not able to improve on hospitalization rate due to volume overload over and above what is currently achieved with other small molecules. But in fact, in key secondary endpoints, we saw some spectacular results. And in particular, we saw a 60% reduction in MACE events, major adverse cardiac events of ischemic origin, meaning reductions in heart attacks and strokes. And notably, in the class 2 patients, we saw a 60% reduction in cardiac death. And just to put that into context, we have identified that patients at greatest risk of progressing to death are those in class 2 with elevated CRP as a biomarker of inflammation. And in our particular study, which was large enough and long enough to be able to look at natural history of patients in class 2 and class 3, we were able to determine that in class 2 disease, which is the major market that most drug companies target because it's the largest opportunity in heart failure, in class 2 disease, even in the presence of advanced disease as defined by high levels of NT-proBNP at baseline, if you did not have elevated CRP, which is about 50% of patients, you had a stable disease course for 4 or 5 years. On the other hand, if you're in class 2 disease and you had an elevated CRP as a biomarker of severe inflammation, then you may have been relatively stable for about 20 months, and then you crashed. And so those patients then crashed to have a high mortality of the order of 30 to 35% over a 3-year period, which at that point parallels the death rate in class 3 disease. In contrast, the single injection of our cells at baseline completely protected those patients with inflammation with CRP elevation, such that for a 4 or 5 year period, they had as stable a disease course as patients who otherwise were not inflamed. And that was a very dramatic effect that in patients with elevated CRP gave us almost an 80% reduction in cardiac death, quite dramatic. And so to be able to identify a biomarker for disease mortality, and also the same biomarker predicts a treatment response, is a very, very unusual thing and goes to the heart of the mechanism of action as well as the potency of the therapy.
I can see that investors might be a bit confused with all this data, some of which seems a bit contradictory. But if you could sum up in just a couple of lines how perhaps they should view the totality of data in your opinion, can you give me that? Sure.
So our cells are very potent anti-inflammatory agents. The heart is the center of inflammation in patients with severe heart failure. And by turning off the inflammatory process in the heart, 2 things are happening. One is we're protecting the viable and salvageable heart muscle from death, and that protects the patient from death due to cardiac causes. And secondly, the inflammatory process emanating from the heart is causing accelerated atherosclerosis in the large vessels outside the heart, the coronary arteries and the cerebral arteries. And again, by switching off the inflammation in the heart, we're able to also stave off those major vascular events. So traditionally, the FDA and other regulatory bodies look at the 3-point ischemic MACE, heart attacks, strokes, and cardiac death, as a composite for risk reduction in patients at high risk of cardiovascular events. And look, we significantly reduced the 3-point MACE as a composite of all of those adverse outcomes right across the 527 patients who participated and received either treatment or sham injection. So I think what we're seeing here is a substantial reduction in ischemic events that I think are going to have a major impact on outcomes in patients with severe heart failure. But I think the implications go well beyond heart failure. Clearly, diabetics are a major risk population. They accounted for 40% of our heart failure population, and we saw these same benefits across the diabetic population. And we will look to expand our focus outside of heart failure into diabetes per se, and of course other patients at high risk of atherosclerotic events would be natural targets for this therapy.
Okay, that's the investors. What about the regulatory agencies? Because you're going to have to do more trials for regulatory approval. How are you going to change them or alter them in the light of what you've learned, particularly about endpoints, I guess?
Sure. Well, look, we hold regenerative medicine advanced therapy and RMAT designation for the product for end-stage heart failure in patients who have an artificial device, an LVAD, in place. And the reason for that the FDA designated the product an RMAT was because of very persuasive data in that patient population where the placement of an LVAD on top of end-stage heart failure creates a massive inflammatory process, the LVAD being a foreign body. And the consequence of that is severe inflammation in the gut and major bleeding events occurring in something like 30% of patients, which cause major hospitalizations and transfusion requirements and a potential for death. In 2 trials now, randomized controlled studies, we've demonstrated that we significantly reduced these GI bleeding events by about 70% as a result of reducing the inflammation in the heart. And in discussions with the FDA have made it clear that they would look at licensure of the product on the basis of a single trial that was well conducted, that demonstrated a cardiovascular benefit. And they've also told us that mortality is the main outcome that they would look for to make a difference in whether or not a product gets licensure in this space. So the fact that we've reduced mortality in this phase 3 trial, I think, is a big deal in a disease state that is driven by high levels of cardiac death, where other drugs don't have such benefit. I think we're looking forward to having those discussions with the agency around whether the secondary endpoints of mortality and reduction in MACE events could support licensure on the basis of the existing data, or do we need one more well-conducted trial that can confirm these results? So we'll be having those discussions in the short term, and we're looking forward to those discussions. At the end of the day, reducing mortality in a high-risk population, particularly when you've got biomarkers and the ability to define the population at risk very precisely, is exactly what the FDA looks for.
And that weird thing that you didn't reduce hospitalizations, which is kind of strange. Have you got a theory as to why that might be?
Yeah, sure. Look, we reduced hospitalizations due to the non-fatal ischemic events. That's clear by 3-4-fold. We didn't reduce hospitalizations due to volume overload. And the volume overload situation really reflects an already damaged left ventricle that is not able to cope well with excessive salt retention. Where our cells don't reverse an already damaged ventricle, that is well established. What our cells do is protect the viable but at risk residual myocardium so that it doesn't die and you don't get further fibrosis and further dilation. And that's what we can do, right? And that's why we think we specifically reduce cardiac death in class 2, but not class 3. The class 3 patients, we believe, are too far gone, too dilated, too fibrotic. The sweet spot for our therapy is class 2, advanced class 2 disease, where there's inflammation, where there's enough viable myocardium that you can protect. However, at that point, patients already have, some fibrous replacement, already have dilated left ventricle. And to think that we can magically reverse the damage, I think was probably going down the wrong path. And I would say, there are plenty of drugs today that improve the handling of salt and volume, and they do a good job. And so I guess thinking about that mechanism of action says that our cells as a single injectable intervention is going to be, they're going to be complementary to the drugs that actually reduce the recurrent hospitalizations from volume overload. Very different mechanism of action, going to be highly complementary. And in fact, in the phase 3 trial, we've looked at the data in that way, and that's precisely what we've seen, that those patients who were on Entresto, for example, alone, did better than those who were not on who were not on Entresto nor our cells. But the combination of Entresto plus an injection of our cells appeared to be highly synergistic. And so we would expect that our cells will be synergistic with any of the other drugs, including SGLT2 inhibitors.
So really, this is an opportunity that is particularly suited to early diagnosis and early treatment. And I was wondering, it's a big opportunity. What are your partnership strategies here?
Yeah, look, I guess advanced class 2 is probably the sweet spot and target market for most heart failure pharmaceutical companies as well as us. It's a very large market opportunity. We can precisely identify the patients who would benefit. It's a single injectable. And so what we clearly will enter into a strategic alliance with a strategic partner that is committed to the space already, where we see eye to eye around the target market, the end user, the delivery to interventional cardiologists, but at the same time, an alliance that's also built around the ability to scale up manufacturing. We have proprietary capabilities to produce the type of volume of product needed for these large numbers of patients. The supply chain logistics need to be built out. And I think an alliance with an appropriate strategic partner will involve all the way from the supply chain through to the commercialization capabilities, which frankly are going to be requiring the kind of investment dollars that existing pharmaceutical companies that have already built out their channels can leverage.
It's a fascinating area. It's really interesting, but you have got other things in the pipeline as well. We unfortunately haven't got enough time to give them as a greater focus, but just briefly tell me about your Ryoncil product.
Sure. So Ryoncil is our product that's been developed for intravenous use, targeting systemic inflammatory conditions. The lead indication is pediatric graft-versus-host disease. We've generated data across 3 different studies, over 300 children, where the cells were used both in the salvage population, children who'd failed other biologics, as well as first line after steroids had failed. And we met the pre-specified primary endpoint in our phase 3 trial, as agreed to with the FDA, single open label study in children who failed steroids. and where the pediatric transplant community could not be persuaded to do a randomized controlled trial. A single open label study was what was in place and agreed to with the FDA. We met the predefined endpoint. We filed a BLA, we had a priority review, and we went before the advisory panel of ODAC last September, and the ODAC panel voted 9-1, almost unanimously, that the data supported the efficacy of the product in children with steroid-refractory GVHD. It's a disease for which there are no approved therapies for children under 12. The only approved therapy in general for GVHD steroid-refractory is Jakafi, and Jakafi has got a lot of adverse events profile in children. That's why it's not approved in children. We have a very safe profile. The FDA acknowledges the safety of the product. And unfortunately, despite a very strong recommendation from the experts, the ODAC panel, the FDA gave us a complete response letter and asked for another randomized controlled trial to shore up the evidence around efficacy. Look, we continue to be in discussions with the FDA, and I expect that we will come to agreement in due course around the ability to have the product approved under accelerated approval. We have given the FDA commitment that we will do a confirmatory second trial in adults with steroid-refractory GVHD, where we intend to show that in advanced disease, grade CD disease, we are superior, both in terms of efficacy and safety, to the best in class, the standard of care. And that'll be the confirmatory study. And we hope to have good news on that front in our discussions with the FDA. Beyond GVHD, we're also evaluating the use of Ryoncil in acute respiratory distress syndrome, another disease of severe inflammation in the lungs this time.
But also, of course, most notably associated with COVID.
That's right. And we had some very exciting pilot data, about a dozen adults at the beginning of the pandemic in New York, Mount Sinai, who received 2 doses of our cells intravenously. 75% of these, 9 out of 12, were able to come off ventilators within 10 days and were discharged. On the back of that, we went into a randomized controlled phase 3 trial of 300 patients in partnership with the NIH in the States. And we recruited 223 patients. We had 3 interim analyses. The first 2 demonstrated a trend in survival benefit. The third suggested that we weren't going to be meeting our high bar of survival with the 300 patients. And so we stopped the trial early. We'll be unblinding the full data set over the next couple of months. And I expect that what we'll see will be subsets of patients where the technology worked well and other subsets where it didn't work so well. It's well established that during the course of our trial, the patient population that was recruited completely changed. So at the beginning of the pandemic, patients who came into the emergency room were rapidly put on ventilators within 24, 48 hours. And we were able to intervene in the ventilated population rapidly when inflammation was still at its peak. I think during the course of our trial, the treatment algorithm has changed such that most patients are treated pre-ventilator with a variety of anti-inflammatory drugs, and almost anything is done in order to avoid ventilator care. And unfortunately, our trial was limited to patients who were on mechanical ventilation. So what we do know is that the second half of the trial recruited patients who were most likely fibrotic, end stage, and had refractory to many therapies, and about 15 years older than the patients recruited in the first half of the trial. So, I think that the evidence with other therapies like anti-IL-6 agents confirms that you need to treat patients, if you're going to be taking care of the inflammatory component, early, not late. And much in the same way as in our heart failure trial, we've learned that intervention with our cells needs to be done earlier rather than later when you've got a fibrotic end-stage target organ. And I think that's what we'll find. And, we've entered into a partnership with Novartis. Novartis is particularly interested in moving forward in a non-COVID ARDS indication. And the objective of unblinding this COVID study will be to identify precisely target populations, biomarkers, etc., where our cells are most likely to work in order to put in place the appropriate ARDS trial that has the greatest chance of success.
Well, there you are. That's the highs and lows, isn't it, of a biotech? But just to finish, just tell me briefly about your cash runway and what investors should be looking for as milestones over the next 6 to 12 months, say.
Yeah, look, we've got about 12 months of cash, and we make sure that we're never that short on cash, that we're less than that. Obviously the FDA approval has been delayed. We had plans to launch the product and had plans for revenues, but that has been delayed by about 6 months now. So we need to ensure that we have appropriate runway. In part, additional cash is expected to come in through our partnerships with Novartis, with Grunenthal in Europe. We expect to enter into additional partnerships in heart failure that I think are likely to increase our revenue. We will have to be managing our cash judiciously, but I think the clinical spend in the next 12 months is going to be substantially decreased since we've completed our current phase 3 trials. We will be focusing on FDA and on partnership discussions as we move both the cardiac and the back pain programs into registration mode.
Silviu, it's been a difficult year for everybody in 2020. How is 2021 looking, and are you looking forward to it? Are you optimistic? Is your glass half full?
Look, as a CEO of a major biotech company, you have to see the world always as half full. I'm particularly excited now because we've put in place large, well-controlled, randomized phase 3 programs, the largest of their kind in cell therapy. And the results have been really nothing short of spectacular. We continue to learn, we continue to optimize, identify where the technology has its greatest strengths, but it's clear that technology works. We have no competitors in the space that we are focusing on. Heart failure, severe back pain, inflammatory lung disease are areas that if we succeed in any one of them, this company will be a major success story. So I'm very optimistic. I think 2021 will be a big year for us.
Silviu, Itescu, thank you so much for joining us.
Thank you for having me.